Carbohydrates, pyruvate, dehydrogenates, and glycolysis. Question 21. Which is not a component of the pyruvate dehydrogenase complex? Dihydrolipoamide dehydrogenase, DLD, is an enzyme that in humans is encoded by the DLD genes. The DLD function as the E3 components of the pyruvate, α-ketoglutarate, and branched chain amino acid, all in the mitochondrial matrix. Therefore, the answer is not A because DLD is considered as a component of the pyruvate.
The question number 21 asks which is not a component of the pyruvate dehydrogenase complex, and the answer choice B, isocitrate dehydrogenase, is actually not one of the three enzymes for pyruvate dehydrogenase complex, so therefore B is the correct answer. The isocitrate dehydrogenase is actually an important enzyme in the tricarboxylic acid cycle, which occurs in the mitochondrial matrix. Isocitrate dehydrogenase is responsible for catalyzing the reverse conversion of isocitrate to alpha-ketoglutarate and the CO2. Answer to C is pyruvate dehydrogenase, and as you probably guess, yes, it is one of the components of pyruvate dehydrogenase complex. It is involved in covalently bonding to pyruvate and liberating the CO2. This is accomplished through its bound coenzyme thiamine pyrophosphate, which is also known as TPP. This enzyme is made in the body from essential vitamin thiamine, which is why many foods are fortified with it.
Because dihydrolipoamide acetyltransferase is a component of the pyruvate dehydrogenase complex, D cannot be the correct choice. Dihydrolipoamide acetyltransferase is the second of three participating enzymes in the conversion of pyruvate to acetyl-CoA, also known as E2. Dihydrolipoamide acetyltransferase functions by taking up an intermediate and reacting with another lipoamide cofactor in order to make an acetyl dihydrolipo intermediate, which releases TPP in the process. Question 22. During glycolysis, isomerization occurs during which of the following reactions? A. Fructose 1,6-biphosphate to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. B. Glucose to glucose 6-phosphate. C. Glucose 6-phosphate to fructose 6-phosphate. Or D. Fructose 6-phosphate to fructose 1,6-biphosphate.
The option A is not correct because the reaction of fructose 1,6-biphosphate splitting into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate is actually the fourth stage of glycolysis. During the fourth stage, an enzyme, aldolase, is used to create cleavage in fructose 1,6-diphosphate, which splits it into two different 3-carbon phosphate isomers. The option B is not correct because the reaction of glucose becoming glucose 6-phosphate occurs in the first stage of glycolysis. During the first stage, a phosphate from ATP is added to glucose, resulting in the formation of glucose 6-phosphate and ADP.
The reason the answer is C, glucose 6-phosphate to fructose 6-phosphate, is because the second step of glycolysis involves the conversion of glucose 6-phosphate to fructose 6-phosphate. This reaction occurs with the help of the enzyme phosphoglucose isomerase. As the name of the enzyme suggests, this reaction involves an isomerization reaction. The reaction involves the rearrangement of the carbon-oxygen bond to transform the 6-membered ring into a 5-membered ring. The reason the answer is not D is because fructose 6-phosphate to fructose 1,6-biphosphate is the third step in the glycolysis. This reaction occurs in the third step of glycolysis when fructose 6-phosphate is converted to fructose biphosphate. Similar to the reaction that occurs in the first step of glycolysis, a second molecule of ATP provides a phosphate group that is added onto the fructose 6-phosphate molecule.